The notes today are about DNA replication, and we are going to talk about DNA replication is, in brief, the copying of DNA. This happens in S phase of the cell life cycle. Okay, remember there's G1, which is growth, then S, which is copy of DNA. Remember, we have to have 2 copies of the DNA for each cell, copy for each cell, and this happens in S phase of the cell life cycle. DNA replication occurs in 3 basic steps. And then I'm going to tell you the steps, and then we're actually going to just do sketches to show you the rest. The first one is to unwind the DNA, and that uses an enzyme called helicase. The second step is to make new DNA, and the key here is that DNA is always made from 5' to 3', and I'll show you that in a minute. That uses an enzyme called DNA polymerase. Your book talks about a variety of DNA polymerases. It's not important that you know that. The third step is to reattach what are called Okazaki fragments, and that uses an enzyme called ligase. Ligase is an enzyme for DNA that makes it attach. All right, I'm going to pause and switch programs a minute. So I'm going to draw the DNA in 2 lines to make it easier to understand. Okay, so normally DNA is in a double helix, but I'm going to draw it like this so it's straight. An enzyme called, now what you need to know about DNA is that this end is called the 5' end. This end is called the 3' end on the same strand. They have slightly different chemical compositions at the ends. That's why, and that's really the only reason you need to know it. We'll talk when we talk about protein synthesis in the next chapter about why else that's important. So this end is the 3' end, and this end is the 5' end. Now, we are making the assumption that you already know that in DNA, A always pairs with T, and G always pairs with C. That's a really important idea. There are 4 base pairs in DNA. So here in our DNA strand, we might have an A, a T, a G, and a C. And on this side of the DNA strand, we have a T, an A, a C, and a G. This enables the DNA to copy itself exactly, and we'll show you that in a second. So the enzyme helicase opens up what's called a replication fork. You don't need to know that. Or a bubble in the DNA. On this side of the bubble, let's say we have something, I'm going to make this very simple and just put little dots in for each of the nucleotides. You can imagine that it's ATGC, ATGC, whatever. Okay? What happens when it opens up as new, I'm going to draw these in green, new pieces of DNA are put in from 5' to 3'. And so A is pairing with T and G is pairing with C all the way along here. Now a little pause. Why? Where do those pieces come from? Where do those extra nucleotides come from? Well, where everything else comes from. The food you eat. Everything you eat is made of cells, essentially, unless you're eating something that's fake, which I don't know that we eat anything fake. And if it's made of cells, those cells have DNA, and there's the DNA you need to put in there. So the DNA forms in a line and forms a new strand, and we have to assume that back here it's already formed, so it's kind of tagging out the other end. And what will happen is the DNA will unwind and split down here, and this line will keep forming all the way down, 5' to 3' all the way. Now what about the other side? Because we've only copied one side of the DNA. I'm going to draw the other side with black dots. And if they're not the exact same number, that's okay. It doesn't really matter right now. Now, the DNA has to copy from 5' to 3'. But the problem is this fork is opening, in this direction, and I highly recommend going to the book website and watching the animation on this. So what actually happens is, and this is really, really difficult to show on this slide in a stationary setting like this. It's easier to show as an animation. That since it's forming 5' to 3', you realize that this bubble is moving again in this direction. So starting here, we have to start here and work backwards and add pieces. So what happens when it spreads open here? Well, then you'll have to start here and work backwards and add pieces. So what happens is when the DNA is put in here, when the new piece is put in, it's put in pieces. The pieces go like this. And those pieces, because, okay, this is laid down in a continuous line. This is laid down in pieces. Those pieces are called Okazaki fragments. Well, those pieces have to be joined together before the bubble closes. And an enzyme called ligase does that. So why pieces? Well, because now what's going to happen is this fork is going to open up over here, and you're going to have to lay down another piece, and then another piece, and then another piece, because it has to go from 5' to 3', even though it's opening in that direction. So what if there's a mistake? This can happen often, especially when you expose your skin to ultraviolet light. You have on a piece of DNA, let's say something like this. Okay, and so the new piece of DNA that's put in would read this: T, C, A, T, G, G, T. Normally, if you're exposed to UV light and your cells are dividing, what happens is UV light promotes what's called thymine dimerization, where it puts 2 T's in for every 1 A. You can see that that would cause a problem. So when our new piece of DNA is made, there are enzymes that go along the DNA. Go along the DNA and read it and look for mistakes. And where they find the mistakes, they cut that piece out. We're not really sure how that works. It's just really cool. So there's a summary of DNA replication.